Hi, my name is William McMichael, and I'm a senior neurobiology major at the College of Worcester. Over the summer, I had a fellowship at Rosalind Franklin University, which is in North Chicago, Illinois. So, as I mentioned in my introduction, I was working at Rosalind Franklin University, which is what's imaged there at the top right corner. And I was not on campus because my work was virtual, but I still had contact with the principal investigator, Dr. Amion Rosenkrantz, whose work is primarily on emotions, taking a circuit level perspective of behavior in rats. And my specific advisor was Dr. Nicole Ferrara, who's postdoctoral researcher there, and a really amazing mentor. So at the beginning of my experience, I was working with photophotometry recordings, which are large data files, unorganized. And so the process of this is to organize this data using a program called MATLAB, which runs on the coding language C++. And that's what's there down at the bottom, a little clip from the files I was inputting into the program to get a more readable version of the data. And here's an example of what photometry data looks like on a readable graph. And this is after processing, so this is a more simplified version of the data I was working with. During the second half of my internship, I was working with a program called ImageJ, investigating fluorescence of neuronal images. This was done through a process called DREAD, which stands for Designer Receptors Activated Exclusively by Designer Drugs. And that's what we have in these images here. The blue is for a nucleus marker of the neuronal cells, whereas the green standing for zinc finger transcription factor. It's a ZIF stain, and that's to indicate learning and cellular expression in the neurons. And the final stain, which you cannot see here, is the stain for the dopamine 1 receptor, and that's a red stain. But here it's difficult to visualize. So by looking at dopamine receptor activation, we were trying to understand how connections between the amygdala and the frontal lobe may differ between adults and adolescent rats. And specifically, we're looking at brain regions, subdivisions in the amygdala and prefrontal cortex, and how these connected. In this slide, I have a little demonstration of the brain regions we were investigating. So on the left there's a little list of the different regions I was looking at, subdivisions in the amygdala and medial prefrontal cortex. And here on the diagram in figure D, you can see the different slices. In A, in the circle, there's the prelimbic cortex, and in B, there's a circle for the prelimbic cortex and the infralimbic cortex. And these were just regions of the prefrontal cortex that are easy to visualize, create kind of a mental image for the kind of connections I was investigating. So as I'm working with fairly large regions of the brain, hundreds and hundreds of images are taken, and this leads to having thousands of data points. And so having this process of data collection being optimized is really essential for timely completion of any work. So a part of this is batch processing, where instead of editing each image one by one, I can take a protocol for editing images and apply it to 100 fluorescent images of the neurons. This greatly speeds up and optimizes the process for data collection. So I think the primary benefit to my learning was just this experience with working with ImageJ, analyzing images, and its relation to neuroscience. So I get a better understanding of the techniques and methods used, like hyperphotometry or DREAD, by just working through the data from them. And these methods are widely used in neuroscience for the circuit level analysis, which is something I plan to do throughout my career. And I also want to put an emphasis on this idea of thinking outside of the box and troubleshooting with these programs and working through the code forces me to understand a problem from a different perspective. I think that's really important, not just in neuroscience research, but life in general. The skills I've learned are very practical for my career going forward as a neuroscientist. Taking a circuit level perspective is something that I want to continue as a researcher, and understanding the tools like DREAD or photometry and how to analyze that data from those tools is something that's going to be a foundation for my confidence going forward as a researcher. I think another important aspect of this internship was that I get to explore a little bit of my passion. And so taking this circuit level perspective has been really interesting to me because it's a nice balance between psychological and strictly behavioral analysis and a really close-up look at like single neurons. So this strikes a nice balance between the two, where you get some involvement with individual neurons and some of the biology, and you get some of the behavioral aspects. I would say one of my key takeaways from my experience is don't be afraid to reach out to people. I think that having that ambition often looks good to the people you're reaching out to, and sometimes it takes a little bit of tenacity to get someone's attention. And I also want to say that this tenacity is not just important for looking for internships, but also within your internship. This summer was difficult for me sometimes being virtual, but even though it can be difficult sometimes, the results are often very rewarding. And that's the end of my presentation. Thank you for listening, and if there's any questions, you can put them in the comments box.